Good day everyone. My presentation today will explore the human gut microbiome, focusing on how it influences health and disease, and the technologies that help us study it. The gut microbiome is an incredible diverse ecosystem made up of trillions of microorganisms that play essential roles in digestion, immunity, and even communication with the brain. However, when this ecosystem is disrupted, it can drive the development of chronic conditions such as obesity and diabetes. By combining our understanding of these microbial roles with advanced research tools like genomic sequencing, we are beginning to develop more personalized approaches to human health. The gut microbiome is a vast community of microorganisms, mostly bacteria. It is estimated that the microbiome contains nearly 100 trillion microorganisms. These microbes provide essential functions, regulating immune systems and strengthening the intestinal barrier. Importantly, the composition of the microbiome is shaped by factors such as diet, antibiotic use, genetics, and lifestyle. Populations in industrialized societies, for instance, often show reduced microbial diversity compared to more traditional communities, which may contribute to differences in disease prevalence. A healthy gut microbiome is critical for maintaining human health. It is not only help us digest and metabolize nutrients, but it also supports human tolerance. It prevents colonization by harmful bacteria and regulates how much energy is extracted from food. Dysbiosis, however, disrupt these functions. It typically involves a loss of microbial diversity, a reduction in beneficial bacteria, and an overgrowth of opportunistic pathogens. This imbalance contributes to systemic inflammation and metabolic dysfunction, driving chronic diseases such as obesity, diabetes, cardiovascular disorders, and fatty liver disease. These effects occur largely through signaling molecules like short-chain fatty acids and bile acids, which link to the microbiome directly to host metabolism and human regulation. Obesity is a complex condition that is not just the result of overeating or inactivity, but is also influenced by genetics, lifestyles, and the gut microbiome. Researchers shows that the individuals with obesity often displays lower microbial diversity and an enrichment of bacteria that promote inflammation. Gut microbes also play a role in regulating appetite and fat storage through their effects on hormones like leptin and GLP-1. Additionally, dysbiosis in obesity promotes the release of bacteria and toxins, which drive low-grade systemic inflammation and insulin resistance, worsening weight gain and metabolic disease. Recent scientific study provides more detailed insights into the microbiome and obesity. A 2023 metagenomic study on obese Chinese children revealed distinct microbial patterns compared to healthy individuals. This obese group had reduced microbial diversity and an enrichment of bacteria associated with inflammation. Functional analysis shows disruption in carbohydrate, lipid, and amino acid metabolism, which can directly influence energy balance and fat accumulation. These findings highlight that the microbiome is not just altered in obesity, but also actively drive its progression. Such microbial signatures could potentially be used as biomarkers to predict obesity risk and to design targeted interventions. One of the most important technologies for studying the gut microbiome today is metagenomic sequencing. Unlike traditional methods that require cultural microbes, metagenomic sequence all microbial DNA directly from samples such as stool. The workflow involves extracting DNA, sequencing, and then using computational tools to identify microbial species and annotate functional genes. This provides not only species-level resolution, but also information about the metabolic capabilities of the microbiome. It has been crucial for identifying microbial biomarkers and understanding the functional contributions of microbes in conditions like obesity. Metagenomic sequencing has been widely applied in research and clinical settings. Projects like the Human Microbiome Project have used it to map microbial diversity across healthy and diseased populations. More specifically, studies have identified microbial signatures linked to obesity and other metabolic disorders. These signatures can act as biomarkers for predicting disease development and guiding early intervention. Furthermore, by understanding the functional changes in the microbiome, researchers can design personalized probiotics, prebiotics, and dietary strategies to restore microbial balance and improve metabolic outcomes. The future of microbiome research is moving towards precision health. Personalized nutrition and probiotics tailored to individual microbiome profiles are already being explored. Fecal microbiota transplantation, currently used for recurrent Clostridium difficile infections, is also being investigated for obesity and metabolic disorders. Advances in artificial intelligence can integrate vast amounts of microbiome data to build predictive models for personalized treatment. Moreover, introducing ancestral-like diversity through high-fiber diets and lifestyle changes may help counteract the negative effects of industrialized diets on the microbiome. In conclusion, the gut microbiome plays a fundamental role in maintaining health by regulating metabolisms and immunity. This biosis significantly contributes to obesity progression by promoting inflammation and disrupting metabolic signaling. Metagenomic sequencing has given us powerful tools to explore microbial diversity and function in unprecedented detail. Looking ahead, healthcare will likely adopt microbiome-based diagnostics and therapies as central elements of personalized medicine. That's all from me. Thank you.